Hello, my name is Ursula Abou-Rjeileh, and today I will be presenting on the effect of palmitic and oleic acids on lipogenesis in dairy cow adipocytes. To start off with some background information, the transition period of dairy cow is known to be critical since the cow experiences sudden changes in metabolic and immune function. During this period, at the onset of lactogenesis, the cow mobilizes fat to meet milk production needs. So lipolysis, which is the breakdown of fat, is favored over lipogenesis, which is the synthesis of fat. And then energy is partitioned towards the mammary gland. However, excessive lipolysis induces remodeling of the adipose tissue, which is characterized by an increase in inflammation and macrophage infiltration. This, and along with a decrease in dry matter intake, intensifies negative energy balance. The negative energy balance puts the cow at a higher risk of developing metabolic disorders like milk fever and ketosis, clinical mastitis, and reproductive disorders such as retained placenta and uterine infections. All of these diseases are costly because they affect milk production and reproductive performance, and they also put the cow at a greater risk in future lactations to develop diseases. So to address these problems, a lot of research has been done at Michigan State over the past couple of years, and researchers have found that targeted fatty acid supplementation in dairy cows has beneficial effects on health and production. In a study done by De Sousa et al., they found that palmitic acid increases energy output in milk, while oleic acid minimizes body weight and body condition score losses and increases plasma insulin. So feeding supplemental fat may be a strategy to increase energy intake and reduce the negative energy balance. In another study, they combined palmitic and oleic acid at different ratios. They had a controlled diet with no fatty acid supplementation, a diet supplemented with 80% palmitic and 10% oleic, a 70-20 blend, and a diet supplemented with 60% palmitic and 30% oleic. They found that fatty acid supplementation increased energy intake, plasma insulin, and milk energy output compared with no fatty acid supplement. And they also found that within the diets with fatty acid supplements, increasing oleic acid tended to linearly increase plasma insulin and linearly decrease plasma NEFA. So now that we know that there are a lot of benefits of fatty acid supplementation in dairy cows, what is the mechanism by which palmitic and oleic acid reduce this body weight loss? So in a study done in bovine adipocytes, it was shown that the effect of fatty acid supplementation depends on the type of fatty acid. This is similar to what we've seen in vivo. In this study, they had oleic and linoleic acid, and they found that these two increased lipid accumulation the most compared with other fatty acid treatments and also compared to control. However, they found that palmitic acid did not differ from control. It is important to point out that in this study, they didn't include the combination of any two fatty acids together, and they also didn't explore specific mechanism as to why oleic acid enhances lipogenesis. So based on these results, we hypothesized that oleic acid alone or in combination with palmitic acid, promotes lipogenesis in cultured adipocytes. Our goal for this study was to evaluate the effect of palmitic and oleic acid on lipogenesis in dairy cow adipocytes. So going into my materials and methods, subcutaneous adipose tissues were collected from 8 multiparous non-lactating, non-gestating Holstein cows, and the tissue was digested using collagenase, and pre-adipocytes were isolated, plated, and induced to differentiate. After induction, mature adipocytes were cultured in standard differentiation media, which was refreshed every 2 days, starting from day 2 until day 14. The treatments were a control with no fatty acid supplement, palmitic or oleic acid alone, supplemented at 100, 200, and 300 micromolar. And we also did a palmitic oleic acid blend, which is similar to what was done in vivo in D'Souza study. We had a 60% palmitic to 40% oleic, a 50-50 mix, and a 40% palmitic to 60% oleic, all supplemented at 300 micromolar. After 14 days of treatments, lipogenesis efficiency was analyzed using adipored, which is a fluorescence-based assay. So tag content was normalized by DNA. And RNA was collected for gene expression using real-time PCR. We also took images of our cells using the InQSight live cell imaging system. Bodipi was used for staining the lipids and NucSpot for nuclear stainings. And then we took images 4 times a day every 6 hours. Our data is analyzed by a mixed model using JMP software. Lipogenesis efficiency is reported as LS mean plus or minus standard error of the mean, and relative gene expression is reported as LS mean in upper-lower confidence interval. Statistical significance was set at p-value less than or equal to 0.05. Now going into my results, as expected, the palmitic and oleic acid treatments alone increased lipid accumulation in a dose-dependent manner. Interestingly, in the palmitic oleic acid mixes, we saw that as we increased the percentage of oleic acid within the mix, we saw a decrease in lipogenesis. Overall, the treatments 300 palmitic, 300 oleic, the 60-40 mix, and the 50-50 mix increased lipogenesis compared with control. Using the InQSight live cell imaging, we could see that even at just 4 days, fatty acid supplementation increases the triacylglycerol, which is represented here in green. And again, this shows that palmitic and oleic acid promote lipogenesis. Next, in order to get a better understanding of the mechanism by which these fatty acids increase lipid accumulation, we measured gene expression of various lipogenic genes. First, we have paralipin-1, which is a protein that sits on the surface of the lipid droplet. So higher expressions of PLIN1 reflect more lipid droplets. We saw that the 200 palmitic treatment decreased PLIN1 expression compared with the 300 oleic and the 60-40 mix. Next, we looked at DGAT1, which is the rate-limiting enzyme for triacylglycerol synthesis. And we saw that the 60-40 mix increased DGAT1 expression compared with the 200 palmitic treatment. Adiponectin is a hormone produced by adipocytes that enhances insulin sensitivity. So we would expect, if we had higher expression of adiponectin, we would expect higher lipogenesis. And we saw that the 200 palmitic treatment decreased adipocay expression compared with control, but the 300 oleic and the 60-40 mix were not different from control. So to summarize, palmitic and oleic acid alone increased lipid accumulation in a dose-dependent manner. The treatments 300 palmitic, 300 oleic, and the 60-40 and the 50-50 mixes enhanced lipogenesis compared with control. And the palmitic acid alone suppressed the expression of lipogenic genes. However, when it was combined with oleic acid, that effect was attenuated. So our results provide mechanistic evidence for the use of oleic acid alone or in combination with palmitic acid in dairy cow diets where fat accumulation is needed to minimize lipolysis. This provides initial evidence for better understanding the mechanism of how fatty acid supplementation may alter energy partitioning in dairy cows. As for our future studies, we're interested in looking at more specific mechanisms as how palmitic and oleic acid affect lipogenesis in dairy cow adipocytes. And we've already identified perlipin 5 and PPR alpha as early drivers of oleic acid during lipogenesis. I would like to thank Dr. Contreras and the lab group for all their help and hard work during this project. And I'd like to also acknowledge Dr. Locke and the CMIB program and the Office of Associate Dean of Research at the College of Vet Med at Michigan State, and also USDA for funding this project. Thank you, and I can take any questions. Please feel free to email me.